The human genome has a complex spatial organization and consists of an extremely long linear arrangement of base pairs that are densely packed inside the cell nucleus. The visualization of this sequence is very challenging due to its hierarchical structure and the difference in size between the cell nucleus and a base pair by several orders of magnitude. Visualizing the different scale levels of the genome data as shown here helps us to understand the spatial organization and hierarchical structure. However, the dense structure leads to visual occlusion. In this previous 3D multiscale visualization, the surrounding structure is made invisible to remedy this problem, and only a maximum of 2 scales are visible at the same time by temporal control. Illustrators have already developed expressive concepts to make the multiscale nature and structural composition across multiple levels of the human genome visible. Inspired by these illustrations, we have developed an interactive multiscale unfolding technique that visualizes several hierarchical scales of DNA. Our contribution is the automatic generation of such a visualization, which combines the individual scales from 3D into a single unfolded view based on real data. The multiscale unfolding technique allows viewers to explore the different scales in one view and to understand how one scale is spatially folded into the next. As you can see, we maintain the local organizational characteristics at each scale and at the same time linearize the higher level organization. We realized multiscale unfolding by defining 2 types of zones, a constant scale zone at each row and a transition zone between the rows. Inspired by the illustrations shown earlier, the transition zones between adjacent scales use the design components of scaling, transparency, and we render the elements on top of each other to represent a containment relationship. We unfold the spatial structure of the genome and start with the individual chromosomes as shown here on top. We can see how our locus, represented here as a single sphere on the causa chromosome scale, becomes more detailed on the loci scale. In the transition zone, we first represent the locus as a single sphere as in the causa chromosome scale. In addition, we render the same locus in the more detailed loci scale to illustrate the folding from one scale to the next. To ensure connectivity, we perform the spatial straightening of the loci scale by calculating a displacement vector based on the positions of the spheres, the previous and subsequent loci in the original data, and use it to reposition this ball between the linearized new positions of the previous and subsequent loci. We change from loci to fibers by connecting the linearized sequences. From the last locus, we can see the transition along a curve and render the fibers on top. Again, we use the approach of spatial straightening. You can also see the increase in size in the transition zone. One locus here contains 100 fibers. In this transition, we can see that the fibers are actually composed of a large number of 100 nucleosomes. Each nucleosome is rendered here in a different color. The nucleosome consists of a double helix wrapped twice around the protein. Because of this inherent structure, we cannot use the approach of spatial straightening before. However, we can unroll the double helix to straighten it. In the final transition, we can see how the helical turns disappear and the base pairs align themselves. To achieve a fully 2-dimensional representation, we first rotate the backbone of the DNA and then flatten the base pair to resemble the chemical formula. In order to depict the several orders of magnitude in scale crossed, we superimpose our visualization with a ruler that schematically show how much length of the detailed scale is represented in the coarser scale. Following the inspiration of traditional illustrations, we have created an expressive multiscale representation of the human genome. However, we also offer the user the opportunity to explore the sequential data while benefiting from the almost complete genome view. We can pan any of the scales beyond the chromosome scale, which makes the remaining scales pan as well at the appropriate higher or lower speeds, depending on the scale difference. Also note that due to the involved rotations, the double helix scale rotates around its axis while panning. And in the transition from double helix to nucleosome, we added a twisting animation. On the top scale, we have the chromosomes, which are not connected with each other. To allow users to quickly jump to different locations, we thus allow them to pan the chromosomes without affecting the rest of the structures, and then to select a new chromosome by double-clicking it. Our multiscale sliding technique allows the user to pan at a speed that is true to scale. We use the Y distance to the first click to control the panning speed. Here we can drag up and float above the coarser scale, thus increasing the gain factor. Similarly, the gain factor decreases as we hover over a detailed scale. Note that whenever we change from one scale to the next, we also flip the direction of the motion. This one-dimensional interaction method allows the user to fluently control the speed for navigation within the extremely long DNA sequence. To get an idea of the surroundings of a particular element at the top 3 scales, the user can view the distances to the other elements. By clicking on a given element, we color map the distances to the elements of the top 3 scales. Overall, multiscale unfolding is a technique that allows the structural composition of DNA from chromosome down to the atomic details to be seen in one view, with one chromosome unfolding continuously to effectively show features in each scale. Both the 3D and the multiscale unfolded visualization helps us to understand the structural composition and the building blocks of life in the human genome.